sometimes there's a combination of these five, so from 1 to 5 of them, that work in concert to control transcription. We talked about activators and repressors and how they can work. Now we're going to talk about how the nucleosomes can be changed to influence transcription. Okay, so we know that chromatin is the DNA wrapped around histone proteins, and they can be a closed chromatin formation or an open, with the closed not allowing proteins access to the DNA, whereas you can see here in the open there's spaces between that proteins can bind in. This is a dynamic process. So there are ATP-dependent chromatin remodeling complexes. So these are proteins whose job it is to change the position of nucleosomes, okay? So these complexes have this DNA translocase that moves along the DNA and that can easily just change from one conformation to another, okay? So the closed conformation to the open conformation and back again. There are several families of proteins known to be able to play this role, and some of them are listed here. So these proteins are chromatin remodelers, okay? So this SWI, SNF, this ISWI, and so forth. These are chromatin remodeling complexes. So how do they change the structure? So sometimes there's a change in the nucleosome position. So what does that mean? We can shift the nucleosomes where they are. So we see here, we have these 5 in a row. We can use it where we open part of it or open space in between all of them. We can kick a histone out. So this is histone eviction, where we are kicking histones out. Or we can change the histone, so we can introduce variance. So take out the normal histones and put in variations of them, okay? So these are 3 ways that chromatin remodelers can change the chromatin structure. Now, I talked about adding histone variants. So there has been variants identified for all histones except for H4. And I'll also note here that when our DNA is replicated and our histones are put in place, they are usually the normal ones. And then the histone remodelers can come back and change them for a variation. So this table shows you the standards and the variants. And I will point out, like here, this macro H2A, this is abundant in inactivated X chromosomes, okay? And then there's this SYNH3 that's found in the centromeres. So it is known that some histones belong to certain areas on a chromosome or certain chromosomes, and we have identified the roles that they play. Now, another way that this can work is by changing the histone tails. So our histones are made up of this globular domain and then this amino terminal tail. And as you can see here, this amino terminal tail can be modified. So here you see some phosphorylation, some acetylation going on, some methylation, okay? So we can modify those tails. And depending on how we modify those tails is going to dictate how our histones can interact with our DNA. And it can also dictate which proteins are attracted to the histones or to the DNA. And by doing this, we can control transcription. So if we remove or change certain modifications here, we can change the charge on it. And by changing the charge, we know DNA is negative. So if we change a positive charge to a negative charge, it's going to have a different reaction or interaction with our DNA molecule. Alternatively, this is like a language that chromosomes speak. So remember we looked at the major grooves and how proteins recognize certain sequences within it. It's a similar concept here. So this can say if we're going to attract activators or repressors to do the job. Now, there is a such thing as this nucleosome-free region. These nucleosome-free regions usually flank a gene, so they occur at around the core promoter and around the terminator site, okay? So this is flanking our genes. It's identifying the beginning and the end. Why might that be beneficial? What is the purpose of having a nucleosome-free region at a core promoter? So think about that and come up with an answer. So we know that this exists, and we know that this is around are in our eukaryotic cells. So we can also change different things here. So we have this nuclear free region, right? And then it has these nucleosomes on either side of it, and it's dictating a start site and an end site. This activator can come in and bind to its enhancer, which can be, remember, near the start site, or it can be at a distance away and physically manipulate the DNA. Well, when it binds there, it can recruit a chromatin remodeling complex. So remember those proteins we talked about that have the DNA's translocases. And their job can be, it can also pull in this histone acetyltransferase whose job it is to modify. And so by it doing this, it can then change this chromatin structure. So it can remove some histones, it can change them, it can replace them. Okay? And so now by it doing that, it's freeing up even more space for our transcription factors and for our polymerase to come in and bind. Okay? And so now, as our polymerase will move along, those histones in front of it have to be removed. So this adds another layer of complexity to transcription. So we have this nucleosome-free region around our start site and our enhancer, our regulatory binding sites. And our activator comes in, and then our activator is going to recruit transcription. It's going to recruit histone remodelers and histone modification proteins, and they then start to change this histone to allow us to get our polymerase in for it to do its job, and then they're then going to move ahead of polymerase, causing the histones to fall out of place so that polymerase can do its job, and then to reform after polymerase does its job. So remember we said our DNA closes back up behind it. Well, it's also going to wrap around the histones. Okay? So I hope this made some kind of sense to you, and we can further discuss it in class if any questions arise.